that. And we're still recording? Yep. Okay. So we're going to finish up today with enzymes. And I still want to keep this, the thing I keep telling you over and over is I want you to start thinking about things in 3 dimensions. And they're not just flat like they are there. They're 3-dimensional things. And in this cartoon, you're seeing where you have a substrate binding actually changing the 3-dimensional conformation of what part of the enzyme? What specific part? Where does the substrate bind? The active site. Now think about that. So if I isolate 100 invertases, for them to be an invertase, which means they have to grab sucrose and split sucrose into fructose and glucose, those active sites have to, almost all of them, be really, really similar in structure. Otherwise, they wouldn't bind sucrose. Okay? Now, we want to talk about inhibitors. And we finished up with this V versus S curve. And now we want to talk about something that's real, and there are molecules that inhibit enzymes. And there's the first class, which we're really not going to spend a lot of time on. These are irreversible inhibitors. These bind covalently to the enzyme and knock it out. It is no longer capable of doing anything. A lot of drugs do this. A lot of drugs inhibit enzymes. They covalently bind to the enzyme, and that enzyme can no longer can no longer recognize a substrate, so no reaction occurs. The ones we're going to spend a bit of time on, though, are inhibitors that are reversible. And there are 2 kinds. They're competitive and they're non-competitive. And we're going to go back to our guy moving boxes on the back to talk about these. And I've told you multiple times that enzymes are incredibly specific. So invertase only sees sucrose. Let me tell you now that we can make a molecule that looks a lot like sucrose that if the enzyme squints and looks at it, it looks like sucrose, but it's really not sucrose. It could be a molecule called sucrose polyester, which is an artificial sweetener that we can't digest. Therefore, we don't get any energy from it. Therefore, it's Splenda, I think. Is it Splenda's sucrose polyester? Splenda, okay, so, and we're going to go through some other real world examples. So now let's go, where it combines at the active site. And what does it do? I'm going to come back to this later. We have the reaction occurring. Here's a substrate, here are the products. Now, in this cartoon, you see this is the true substrate, but the inhibitor now in this cartoon, instead of being round, is square. So it actually binds, so it still has the 3-dimensional geometry of the true substrate, but nothing happens, so it just spits it out. Okay, so when this occurs, no products are produced. Is that clear? Okay, let's go back to drawing the guy on the loading dock. And we're going to put substrate and velocity. And here's what we got the Wednesday. Now, let's add a competitive inhibitor. So we have the guy on the loading dock that moves boxes, and he moves a certain kind of box. And our competitive inhibitor, let's say his job, his enzymatic function is to take the box, move it inside, and take the lid off. And we determined he could do, 4 boxes a minute or whatever it was. So let's give him a competitive inhibitor. A competitive inhibitor would be a box that doesn't have a lid. It's just a box. So he picks up the competitive inhibitor, takes it inside, can't take the lid off. So what does that do to his speed of moving boxes? Goes down. Okay? So if we have one box with a lid and one box without a lid, and then we're going to have, we're going to go to 2, and let's say this is 1, so he's only going to be able to move 1. Let's say a 4, then, he's only going to be able to move 2. Okay? Do you follow the logic there? Do you follow the math? Is there any way, anything I can do to be sure that this one guy almost always moves box with lids? What's the easiest thing that I can do if I have an unlimited number of boxes? What can I do to make sure that he's always moving boxes with lids? Nobody? Think about it.
Don't ship boxes without lids?
Don't ship boxes without lids? That's without the inhibitor, though. How can I overcome the boxes without lids? Let's say there's 10 boxes without lids and there's 10 boxes with lids. That's a 50% inhibition. What can I do to make sure he never moves a box without a lid? You can question? Yes, ma'am. Right kind of fit? The answer is put 1 million boxes with lids in the back of the truck. So let's say I have 10 boxes with lids and 10 without, okay? We'll go over here. So he's still going to be, let's say, at 5. Now, what if I change the ratio? Here we have 10 to 10. What if I go 1,000 real to 10 without a lid? What's going to happen then? He's got a much higher percent of every time he grabs a box, it's going to be a box with a lid. Is that clear to you? Does that make sense? So if we move our curve out here, what we actually see then is we change the half maximum velocity, but we have the same V max. That's supposed to be MAX. Can't even spell. Okay? Opens up the back of the box, the truck, and there's 1,000 real boxes in there, and there's only 10 boxes without lids. So what's the probability of him grabbing a box without a lid? 10 in 1,000. Or 10 in 1,010. What's the probability of him grabbing a box with a lid? 1,000 out of 1,010. Okay? So we can overcome competitive inhibition by adding more substrate. True substrate. The boxes with the lids are the substrates. The boxes without the lids are the fake substrates. We have a tremendous amount of research trying to do this with drugs. That's what drugs do a lot of times. And what we do is when we take a medication, we're giving that enzyme a tremendous amount of the fake substrate. So the enzyme doesn't work. Okay? Does that make sense? Now let's talk about a non-competitive inhibitor. And let me first go back and show you again the competitive inhibitor binds at the active site. So a competitive inhibitor, and I like to say if the enzyme squints and looks at the competitive inhibitor, it looks just like the substrate. So cyanide looks just like oxygen. But you don't want to breathe cyanide or eat cyanide because any enzyme in your body that sees oxygen is going to see cyanide. So hemoglobin, instead of carrying oxygen, is going to carry cyanide. That's why it's so fast. But even if you consume cyanide, you've got a few seconds. It's dispersed in your body. It kills you several different ways. But it all has to do with the fact that the enzymes or hemoglobin, which is not an enzyme, but it still does the same thing, sees cyanide. And you can only survive if you get a really, really small concentration of cyanide and you happen to be at a hospital where they can give you another competitive inhibitor that can outcompete the cyanide. And they'll also maybe, they'll put you on an oxygen, on pure oxygen, so you're increasing the amount of oxygen. Okay? Non-competitive inhibitors bind somewhere else. Okay? The big difference here is this non-competitive inhibitor right here binds to some other site on the enzyme, and when it does, it changes the shape of the active site. It warps the active site, so now the active site can't bind the enzyme. The substrate, I'm sorry. The substrate as well. Does that make sense? So the reaction rate goes down. Okay? So if our guy is moving boxes, a non-competitive inhibitor would be someone that comes up and stands behind him and grabs his arms. So he goes in to get the box. He can't grab the box because the non-competitive inhibitor has bound to his back and is grabbing his arms and moving his arms so his arms don't have the right three-dimensional shape to grab the box. And there's really not much you can do about that. Okay? They will release and combine, but so if you had a lot of substrate of true boxes, it's not going to help him any at all because he can't grab the boxes. You can add as much substrate as you want. It's not going to change. Does that make sense to you? You know, you've warped out the active site. The active site was his hands held in such a way that he could fit the box in there. Now his hands are, held over his head or whatever. And so, just very quickly, if this is our normal, then this is our non-competitive inhibitor. We do not get the same Vmax. Okay? Questions? So in the case of competitive inhibitor, we get the same Vmax. If I add 1,000 boxes in there, there's a good chance that every time he grabs a box, it's going to be a real box. In non-competitive, doesn't matter. His hands are tied behind his back. His hands are warped out. He can't bind the box, so he can't move the boxes. So non-competitive inhibitors reduce the activity of the enzyme. And the big difference, well, there's several differences, but a non-competitive inhibitor almost always binds at another site on the enzyme than the active site. Okay? Okay, let's talk about a inhibitor that is out in the real world. And this is the shikimate pathway. And we actually have a molecule that inhibits this pathway. And it actually is this step. This is, by the way, if we go through this all the way down, we get amino acids produced. So don't worry about just this is the shikimate pathway. And we have an inhibitor, a competitive inhibitor, that will keep this reaction from occurring. So what happens if this reaction doesn't occur? What happens to the production of amino acids? Production of amino acids goes down drastically. What is the result of that in the plant? Plant dies. If we inhibit amino acid biosynthesis, then we're not going to make proteins, and without proteins, there are no enzymes, there are no structural proteins, there's no integral membrane structural proteins, and the plant dies. Does anybody know the name of the chemical that does this? You all know it. You've all used it. It's a chemical called glyphosate. What's glyphosate called? What's the trademark name for glyphosate? Roundup. Glyphosate, which is the chemical name, inhibits amino acid production in plants. And it does a really good job of inhibiting amino acid biosynthesis in plants, and the plants die. Glyphosate also, when it hits the ground, it's pretty well tied up, so it doesn't run off and kill algae in streams most of the time. It's a pretty good herbicide. There are some problems, and you know, and we will talk a little bit about this later, now you can get glyphosate-resistant cotton, Roundup-ready cotton, Roundup-ready corn. What else? Any others? These have been genetically engineered so that they still produce amino acids in the presence of glyphosate, but the weeds don't. Unfortunately, now there are Roundup-resistant weeds. And that's becoming a fairly large problem in the Midwest especially, because when this started happening, most farmers no longer had tillers. They didn't have a way of going in and tilling the weeds out. So they had to go back to what they used to do a long time ago, which was to hire high school kids in the summer to go hoe the corn. So now what Monsanto is introducing is 2,4-D ready crops, because 2,4-D now they're developing crops that are resistant to 2,4-D. But let's go to another example that I told you you'd be seeing over and over again. Here's the real world reason that plants are affected by oxygen. We had this the first day, and this relative rate of CO2 uptake and oxygen concentration. So we know right here that oxygen is about 21%, okay? So we get this rate of photosynthesis. With 73 parts per million CO2, we get this, 275 parts per million CO2, we get this rate. Our CO2 up is now, what is it, 330 or something? Anybody know? 400? Wow, so we'll be getting up here somewhere. But you will see when we talk about photosynthesis, if we add oxygen, the rate of CO2 fixation goes down dramatically. That's because oxygen is a competitive inhibitor for ribisco, which is the enzyme that fixes carbon. Why? CO2, the enzyme squints its eyes and thinks that an oxygen looks like a CO2. And oxygen binds, a different product is made, but the big deal is you do not get CO2 fixed. And this is real world. This is why most plants don't grow faster than they do. And you can add CO2 to greenhouses. Why can't you add CO2, supplement CO2 in greenhouses in Texas? Too hot. You'd have to have a sealed greenhouse. And we'll go over and if you're up in Michigan, there's some instances where they can add CO2 to a greenhouse and increase the growth rate of, and you guys all know what C3 plants are and what C4 plants are. You've had this before. C3s are the ones that are really affected by oxygen in the real world. So as the level of CO2 goes up, those C3 plants we thought would grow faster. They may not for other reasons. Okay? Questions?
What's the inhibitor in spike 80?
Spike 80, I have no idea. I don't even know what that is.
Spike 80, it stops the photosynthesis process in plants. It's an herbicide.
Spike 80 is a new one? I don't know. Anybody know? If I knew what the chemical was, I might be able to tell you.
I'll look it up.
There's some good ones. You know, the one that you'll get will be paraquat. Anybody ever heard of paraquat? Paraquat was used, it works fast. You can spray it on and come back, and it causes basically, the plant still absorbs light, but that light energy is used to rip the plant apart. And years ago, before any of you were born, we had a program of spraying marijuana in Mexico with paraquat. Unfortunately, they harvested the marijuana and still sent it north, except now it was completely laced with paraquat. And so they had to stop that program. Okay, questions about this? Like I said, you saw this the first day of class, and you'll see this curve a couple of more times. This ribisco is the most, and I'm just telling you this now, this is not going to be on the first exam. By weight, it's the most common protein in the world. Almost all the protein in a leaf is this enzyme, because leaves have to fix a lot of carbon. Okay, let's move on a little bit. Okay, what we're going to talk about now is a very quick overview of molecular biology, of how things work, and we're going to finish up with everything that we have covered so far, and that is finishing up with how bourbon is made. Everybody knows that, I'm sure, everybody read The Double Helix? really short book about the discovery of DNA, Watson and Crick, and a guy named Maurice Wilkins won the Nobel Prize. The person that did a lot of the work was Rosalind Franklin, and Rosalind Franklin did not win the Nobel Prize. Does anybody know why? She died. They do not give the Nobel Prize out unless you're still living.
In all my previous science classes, they always told me it was really good.
No, she died. They don't give it out. She died during this during the period when they were actually publicizing the stuff. And then, if you read the book is really funny, and they did not like her very much. So after she passed, Watson rewrote a new intro to the book. The book's called The Double Helix. So you can read it in an afternoon. It's really fun book to read. And there's Maurice Wilkinson. So the central dogma, and that is that you guys know that DNA is used as a template that produces RNA. By transcription, then RNA moves out of the nucleus into the endoplasmic reticulum for the most part, where proteins are made. But now you know we can take RNA in the lab and we can use an enzyme called reverse transcriptase. You don't need to know that. And we can take RNA and make DNA. And basically we can do about anything we want. And still most of the money in research is going to looking at what gene, the structure of genes. Now, again, one thing you're going to see is that it's just not simple RNA and then messenger RNA. Each of these are a series of incredibly complex steps that involve an enormous number of proteins. We're going to talk about RNA polymerase when we talk about gene regulation, but Dr. Kyle in our department works on one protein that is needed for transcription to occur, and there may be 100 proteins that are needed before transcription can actually start, and we're not going to talk about that. But these are very, very complex processes. I'm going to go through this really quick because you've had this before. A always binds to T, G to C, and if you go back when the way that Watson and Crick figured this out was they made cardboard cutouts with the relative size of the nucleotides. And they realized that for them to get the same outside size, that if they put them together with A with T and G with C, then they would bind together and produce the same size outside, a helix. And so they literally did it with cardboard. Anybody ever heard of Linus Pauling? Linus Pauling, who won 2 Nobel Prizes. He was the guy that figured out the structure of proteins. He was sick one day, and he was sick, and he was reading detective novels, and he finished the novel, and he didn't have anything to do, and he said, okay, well, maybe I'll figure out the structure of proteins. Now, he was a protein chemist, but he got some pieces of paper and drew the peptides, or the amino acids, on a bunch of pieces of paper, and figured out how they went together. You know, no high-tech, three-dimensional modeling or anything, just pieces of paper. And his work was not actually sponsored. He didn't have a lot of U.S. Government support. Who would provide a lot of money for the structure of proteins? What company, commercial company, would be interested in that? A company called Revlon. Because if you're going to modify proteins, you have to know what the structure is, and every time you walk by where someone's having getting their hair curled, you're smelling protein disulfide bonds be broken and be rearranged. You know, because your hair is protein. So Revlon actually still does fund a lot of work. The main difference between DNA and RNA is, of course, that the DNA is a double helix and it stays in the nucleus. The RNA is produced in the nucleus and, of course, travels into the endoplasmic reticulum or the cytoplasm. So, if we look at this again in this cartoon, we have replication, we have transcription, then something we're going to talk a little bit about is RNA processing, and then the RNA goes through the nuclear pore in the membrane, and it either is translated in the cytoplasm or most probably on the endoplasmic reticulum. Now, this is something. This is by the way in one of those chapters I put online. This is something to me. When this first came out, this was like incredible. What it turns out is that in plants and in higher organisms, that a gene, and when I'm talking about a gene, I'm talking about something called a structural gene, which produces a protein, that you remember that RNA is made, okay? Now, in this piece of DNA, there are distinct areas called exon and intron. Exon and intron. Exon, and these will go on and on. What is remarkable is that first, this RNA is made, then these pieces, these introns, are cleaved out, and then the pieces are put back together. So when I took this course, RNA was made. Now we know that this pre-RNA is made and that pieces are snipped out and then the RNA is stuck back together to produce the messenger RNA. That's incredible. Think how complex that is. Every intron has to come out at the right base and all those pieces have to be stitched together correctly. And you can have 12, 15 introns and exons in a gene. Okay, do you understand that? We just don't make a piece of RNA, a messenger RNA. We make this piece of RNA, and then enzymes have to take it and snip out the pieces and put the ends back together again correctly every single time. Okay? And this is one of the steps in what's called RNA processing. Now, I'm going to end on this one because this is another cartoon basically showing you the same thing, and we'll be talking about a couple of other steps here. So we have this transcription of this intron and exon piece of RNA. Then we have the introns removed, and then this piece of RNA has what's called a cap protein added, and then at the other end has a bunch of A's, anidines added to it. Then it has to move through the nuclear pore. Remember the nuclear pore. Then it can be translated. So that's a lot to happen. Okay? So to go from the DNA to the protein, that's a lot of stuff. It's just not transcription translation. Every step, there's just dozens of steps between those two. And everything has to happen right. Okay? Questions? Okay, so you got, what is that, Friday? See you on Monday then. Okay, why can't I see the end? Ah.